Hello, everyone. I'm going to share my screen to introduce this webinar. Wait a second. So today we have 4 talks. The first one is from Jorge Meave, Dr. Jorge Meave. The second one, Ella Vazquez. The third one, Ricardo Rivera, and the last one, Pedro Luna. I'm going to start presenting Jorge Meave right away. And I'm going to allow myself to let you know that he is the reason that I became a biogeographer. He is an excellent teacher, but at that time he made me realize the importance of the wonders of doing biogeography. Last year, he won the National University Award, and his curriculum is just amazing, truly amazing. He's a great teacher, and I hope we enjoy your talk. Thank you, Jorge. Thank you for accepting this.
Thanks, Leti, for your beautiful words. I didn't expect them, but thank you very much. So, good morning, everyone. I also welcome you warmly to this webinar. I think I'm going to stop my video because yesterday we had a really bad rainstorm and my internet is kind of shaky, so that I can share. But first, let me share my screen with you. Let me see if this works.
Perfect, thanks.
Okay, well, first of all, I want to thank Ella Vázquez, Fabricio, and particularly Leti, my dear Leti, for organizing this seminar, and everybody involved for having invited me to give this presentation. I really hope that everyone will have a very enjoyable morning with this series of really interesting presentations that we have today. But particularly in my case, I really hope that through my talk, I will convince you that Humboldt was indeed the first formal ecologist of modern times. So first, I will explain to you what the outline of my structure of my presentation will be. I will first briefly review the definition of ecology as a biological science, and then I will provide a quick overview of Humboldt's contribution to the study of plant geography and ultimately ecology. Then I will tell you a tale of one volcano based on Humboldt's expeditions to Chimborazo in Ecuador, to finally discuss with you some implications of his work for our current efforts to understand and document climate change. So let's begin with the definition of ecology as a scientific or a biological discipline. To understand what ecology is, I would like to recall that this concept was coined in 1866 by the famous zoologist Ernst Haeckel. This is an important piece of information because it implies that Humboldt had already passed away when the concept was introduced in the science of biology, so that it was impossible that he was called by anyone an ecologist. As you all know, Haeckel combined two Greek terms, oikos, to signify home or surroundings or environment, and logia, to indicate that this is a field of knowledge. Thus, ecology can be defined as the study of nature, the home of all living things on Earth. So the goal of ecology is to examine and really ultimately to understand the ways in which living organisms interact with the environment and with each other, as this interaction drives their abundances and distributions. So those of us who are devoted to ecology recognize in the environment that there are two components, an abiotic and a biotic component. The abiotic component refers to all physical and chemical factors of the environment that affect the lives of living organisms, of which I have listed here some examples. Collectively, we refer to these factors as resources and conditions. In turn, the biotic component refers to the different ways in which the activities of some organisms affect others, either positively or negatively, And we refer to these as biotic interactions. So with this common ground, we can move on to the next part of my presentation, which is related to Humboldt's contribution to the study of nature. So Friedrich Wilhelm Heinrich Alexander von Humboldt. He must have been a very smart person in order to be able to remember this very long name to begin with, I think. An important aspect of his biography was the time he attended the University of Göttingen, which was a very prestigious Prussian institution at the time and still is. And he was formally trained there as a mine engineer. He received a very good education in mathematics, geography, natural sciences, chemistry, botany, and with time his knowledge became so vast that he can be considered a true polymath. For those who speak Spanish and don't know the word, it means erudito. In particular, Humboldt is regarded as the father of plant geography, a discipline with a bicentennial tradition that includes many important figures of ecological botany, such as August Grisebach, Christian Ronquier, and Frederick Clemens, among many, many, many others. So thanks to the understanding that Humboldt had on the role of the environment and the distribution of organisms, he was a big promoter of the use of instruments to measure the physical environment. So what he did is he went out and purchased every possible instrument that was available at the time, including barometers, like these, sextants, thermometers, and even this intriguing device here called cyanometer that he used to measure the blueness of the sky. All these instruments were an important part of the luggage that Humboldt carried in all his expeditions. So this is the reason why many of his works are full of ecological content, although he also wrote about many other topics, including the social and economic structure of the territories he visited, and even the importance of human freedom. One aspect that I want to highlight here is his contribution to the growth of the conceptual framework of natural sciences, as he created many terms that we are still using in our current scientific language. Some examples of these include plant association, isotherm, isocline, Jurassic, magnetic storm plant formation, etc. And in particular, he introduced the method to describe vegetation based on its physiognomy. And finally, I must also mention one important aspect of his scientific thinking, which is that it was holistic. And this is expressed very well in his famous quote, "In this great chain of causes and effects, no single fact can be considered in isolation." At this point, I have to confess to you that in my imagination, he first expressed this idea in the middle of the rainforest to his travel companion and colleague, Aimé Bonpland. And look at his face. He said, "Oh, that's profound." So now I will tell you the tale of one volcano. Chimborazo is a prominent feature of Ecuador's geography, for those of you who have seen it. It is located almost just at the equator. I want to remember that Ecuador means equator in Spanish, but with a towering elevation at its summit of more than 6 kilometers, this place doesn't remind us very much of a tropical or an equatorial location. There is very little doubt that this is the very same house located in Antisana that Humboldt, Bonpland, and their group of assistants used as headquarters for their expeditions to the high volcanoes located in this region, particularly, but not only, the Chimborazo. Climbing these tall volcanoes must have been very difficult without the proper equipment, and there is evidence that at least in one climb, Humboldt fell ill because of the rare air at this elevation. However, his determination led him to conduct many expeditions, and in each of them he collected plants, he made a lot of measurements with his instruments, and he made numerous notes on the distributional ranges of the plants that he encountered. So, among the many products of these efforts, his tableau physique des zones de épis voisins, which accompanies their amazing essay on the geography of plants, I think that's what it's called in English, is perhaps one of the best known ones. In my opinion, it is not only beautiful, but also impressive for all the ecological and geographical information it contains. So through this piece of work and many other similar ones, Humboldt left us a legacy that represents a wealth of ecological information related to elevational gradients in graphic, tabular, and written form. You can see all the amount of information that is written here in a way that we don't do it now any longer, but it was very important. I will tell you why in a minute. So the information collected and systematized by Humboldt and Bonpland represents, in my opinion, what seems to be the earliest account of the elevational distribution of plants in tropical mountains. Among other things, at that time they concluded that the upper limit of spermatophytes was roughly at 4,600 meters elevation. That there were two clearly distinct plant associations with these elevational ranges. But most importantly, the main conclusion of his work was that climate was the primary control, or climate is the primary control of the altitudinal distribution of vegetation. So with this information, we may ask, how do these results contribute to our current analysis of climate change? So we can move forward now, and let's talk about the implications of Humboldt's work in tropical mountains for this purpose. First, I hope that we all are aware of the magnitude of the human footprint on our planet. The transformation that we are witnessing at present are the direct result of our activities, including urbanization, land use change, industrial development, mining, etc. But one that is particularly worrisome is the modification of climatic patterns, because this may potentially affect, among other things, our ability to produce food and the geographical ranges of many diseases. So therefore, how can we produce evidence of climate change and its consequences on vegetation? In trying to answer this question, I think it's important to remember that vegetation is not an unchangeable feature of our planet, basically because it's tightly linked to climate. Both paleoclimatology and historical records have provided ample evidence for climatic instabilities in the past. For example, in recent times, let's say the last 1,000 years, human beings have experienced, two periods that are well known of climatic anomalies, quote, quote, namely the medieval warm period in the Middle Ages, with a peak in temperature at around 1,000 years before present, and the famous Little Ice Age, a period in which agricultural patterns were so deeply modified that probably it also affected the patterns of natural vegetation. So clearly the evolution of human species has taken place in a suite of different environmental scenarios. Indeed, we have coexisted with many species, plant communities, and general with ecological systems, unlike those we know today. In the context of the present climate change issue, in 2015, a very relevant paper came out that used information produced by Humboldt in an attempt to produce long-term evidence of the consequences of climate change and vegetation based on the temperature gradients existing in tropical mountains. This woman, Naya Morueta Holm, and a group of colleagues returned to the Chimborazo 210 years after Humboldt did, and perform a systematic and very careful survey for plant species and communities in order to determine if the original elevational ranges had suffered any changes over the last 2 centuries. I can really not go into the details of the study right now, but believe me that they found very strong evidence that the ranges of the plant communities described by Humboldt and Bonpland had changed a lot. In the case of the Gentianes Association, this is the range described by Humboldt. So this community had shifted upwards quite a bit, while the pajonal vegetation, compared to the original range described by them, had expanded both upwards and downwards, and other species have also shifted upwards. On average, it seems that vegetation had moved on average 500 meters upwards. So when they analyzed individual species, they were also able to identify that 51 of them And for this species, they found enough information to allow comparisons of historical and modern ranges. And this is what they got. For these 61 species, the black bars indicate the overlap of elevational ranges. Red bars indicate contraction of upper and lower elevational ranges, so the species are not occurring here any longer. But the green bars indicate the expansion of the upper range limits. And you see there's a lot of green here. So this means that many species also moved upward, at least most of these 51 species. So based on this information, Naya and her colleagues produced this beautiful update of Humboldt's Tableau Physique, where they show shifts in vegetation and the limit of the glacier and indicate the relative role of climate and land use here in this modification. Climate all over the gradient and land use mostly in the lower elevations below 3,800 meters. So this looks like the end of a nice story, but is it really? Let's recall that Naya Morueta-Holm and her colleagues concluded that their findings provide evidence that global warming is strongly reshaping tropical plant distributions. And yes, I think it's true that their findings are very conclusive in this regard. However, we also must remember that Humboldt climbed the Chimborazo in the early years of the Industrial Revolution, just at the end of the Little Ice Age, that is in the late 18th century. So actually, this is not the end of the story, and on the contrary, the plot thickens. In 2019, another relevant paper came out published by Alexander Koch and his colleagues, in which they examined the effects on Earth's climate and of the great dying in the Americas after the arrival of European conquerors. Actually, this is an issue that has been investigated profusely since the 1960s, and previous studies had concluded that the reduction in population size in the Americas had not had a strong climatic consequences. Yet it's clear that the period where this population reduction took place, which is indicated here by the yellow rectangle, called the Great Dying, coincided with a CO2 anomaly and an increase in photosynthetic activity in the planet indicated by a change in carbon isotopic ratios characterized by a relative increase in the 13 carbon isotope during this period. Thus, what Alexander and his colleagues did, they went on and compiled and critically examined every piece of information available on 4 items. The first is pre-Columbian population size, the second is the per capita land use, the third is the post-1492 population loss, and finally the resulting carbon uptake that took place in abandoned landscapes. So the first question is, why did so many people die? It has been long known that native populations were affected by several diseases that were carried in the biological luggage of the conquerors and colonizers, and that these diseases caused epidemics that affected large areas that were heavily populated, like here in southern Mexico, northern Central America, and the Inca Empire in the 15th century. In 2 of these densely populated regions, namely Mexico and North America, and the large territory occupied by the Inca Empire in South America, these epidemics resulted in incredibly high mortality rates. A single disease, so far not completely identified, and that was called cocolitzli in the New Spain, wiped out from the territory up to 17 million people in 2 outbreaks separated by 3 years. Along with the previous smallpox epidemic, population in what is Mexico today decreased from nearly 22 million to a mere 2 million people in a little over half a century. So overall, the evidence compiled by Alexander Koch and his colleagues indicates a 90% reduction, represented by the death of 55 million people in the Americas, which implied that 56 million hectares were abandoned and secondary succession started. And the consequence in the atmospheric carbon balance was tremendous. This implied a 7.4 picograms removal of carbon from the atmosphere and transfer to the land. And this is equivalent to 3.5, oh, sorry, there's a mistake here, it's 3.5 parts per million reduction in the atmosphere. So all in all, according to their calculations, these changes accounted for at least 50% of the observed decline in the atmospheric CO2 during the 16th century. And it's important to mention here that this group of researchers were careful enough and examined other potential causes of CO2 and temperature anomalies at the time of the Great Dying. Yet they proved that they are unrelated to anomalies in total solar irradiance, as can be seen here, and to aerosols from volcanic emissions. So this was not a result of these events, but it's coincident with an important reduction in land atmosphere carbon fluxes, which in turn are related to a decline in land use change in America, but not in Europe and Asia. So they were able to conclude that the great dying in this continent and the consequent abandonment of land had a tremendous impact on atmospheric carbon and air temperatures in the 2 centuries that followed. So at this point in time, we need to ask again, what exactly did Humboldt as well as Naya Murieta Holmes see in their expeditions to the Chimborazo? If the great dying triggered the little ice age, and this temperature anomaly only ended with the arrival of the Industrial Revolution, it is likely that the elevational distributions of the plant species and communities recorded by Humboldt and Bonpland were an ecological, geographical phenomenon caused by a temporary planet cooling. Therefore, in my personal opinion, we need to be careful as the results of the 2015 study do not necessarily prove current warming of the Earth's atmosphere. So it really looks like we are far from placing the final piece in this puzzle, but I'm convinced that without Humboldt's insight, curiosity, and tenacity, and of course without his data, we might not have even started to solve it. To finish my presentation, I want to share with you my conviction that the impressive progress made by ecology in the last century and a half is basically thanks to the fact that we are riding on the shoulders of Alexander von Humboldt, paraphrasing Isaac Newton's famous quote. And this also applies to my own work, which has been much inspired by the amazing ideas, the profound interest, and particularly the love for nature that characterized Alexander von Humboldt, undoubtedly the first ecologist of modern times. And with this I finish, and I thank you all very much for your kind attention.
If you are up to, we can ask you from the audience a couple of questions, and then at the end, we will allow more people to ask. Well, you can ask the question, but then we can do it at the end. So the first question is from Ignacio March, and he asks you, are you agree to apply in Mexico assisted migration practices to conserve flora and fauna of mountain ecosystems in order to enhance population movement to higher altitudes in the context of an accelerated climate change context?
The first part of the question, the audio was kind of broken and I wasn't able to follow just the first part.
Assisted migration practice of flora and fauna.
If I think that that can be done?
Should be done. I think the question is if it should be done.
Yeah, probably, but I don't think that's easy. I think it's very complicated. But maybe yes. We know that there will be elevation shifts, of course, but we are not still exactly how strong. Based on what I said, we are still uncertain. And it's likely that those communities or biotic groups that are restricted to the high elevations will be those that suffer the most from increases in temperature. And I don't think we know how to assist those, because above the top of the mountains, there's only the sky of the species or the heaven of the species. So yeah, that's a very tough question. Maybe we should also think on latitudinal movement assistance, not only elevation.
Yeah. So we have another question from Diego Santiago Alarcón. Why Europeans did not suffer from native diseases in such a way that could represent a weapon of resistance for American culture?
Yeah, I don't know. I have no idea. I have no information to answer that question, but definitely that's a good question. Maybe it has to do with the fact that the number of people that arrived were much smaller, and but no, I don't know.
Okay. And the last for now, is from Silvia Torres. Jorge, do you know if there are estimations of how this actual pandemic has affected climate change? Yes, climate.
I don't think that has been estimated yet, and I think there are some people interested in that issue, particularly if the patterns of movement, air traveling, and all that implies that we are reducing our consumption of fossil fuels. Maybe there will be a positive result in terms of climate modification, but that also has to be put in terms of long-term analysis, because these changes may be temporary and maybe they may not be enough to revert trends that originated in the last century and are very strong and very difficult to stop.
Yeah, well, very interesting. Thanks again, Jorge, for these amazing presentations, as always. I'm going to give the micro to Fabricio so he can introduce ella Vazquez, and we can continue with our webinar. Thank you, Jorge.
Bye.
Great. Thanks, Jorge, for the great talk. Thanks, Leti, and thanks everybody for being here, being interested in our seminar. Our next talk, I have the pleasure to introduce my longtime friend and colleague and mentor, actually, also, when I started studying biogeography. She's ella Vázquez-Domínguez. She did her bachelor at National University of Mexico at UNAM, and he did also her PhD at UNAM, where she worked with ecology, genetics, and physiology of small mammals. And then she wanted to broaden her scope, and she went all the way to Brisbane, Australia, to the University of Queensland to do a postdoc with Dr. Craig Moritz. And then she came back to Mexico. Fortunately for us, she came back to Mexico, and she was hired at the National University of Mexico, where she is now a full-time professor and leading the lab of genetics and ecology. Her research interests are wide, including from molecular ecology, population, landscape genetics, genomics, phylogeography, hybridization, invasive species, and conservation of endangered species and natural resources. She works mostly with vertebrates, but she also has some interests in other organisms. And it's my great pleasure to introduce Professor ella Vazquez, and she will show us a little bit of her research under a Humboldtian view. Please, ella.
Thank you very much, Fabricio. Very much appreciated. I couldn't, in my wildest dream, think that we will have over 260 participants. I'm just amazed. So let me share my screen and get right into it. As we just brilliantly learned from the previous talk, Alexander von Humboldt, a baron, is no doubt a founder figure of modern geography, famous for his studies of, famous for his studies on theoretical essays about elevation and latitudinal gradients. But however, there has been less attention historically for his pioneer work in landscape archaeology, as described by Ken Mathewson in the Journal of Geography in 1986, where he said that Alexander was an early landscape archaeologist, and he wrote, in effect, Humboldt founded New World landscape archaeology with his studies of Andean and Mexican antiquities. So interesting, we can think of another Humboldt's legacy, a kind of archaeological gradients, stratigraphic gradients, from where we can study how environmental changes and biogeographic barriers that have defined the distribution of biota in ancient and modern times. And before I continue with what the theme of this talk, I really have to emphasize that all of it is the work of an incredible team of former students, postdocs, colleagues, technicians from different fields, from archaeology to forensics, to molecular genomics, ecology, conservation biology, and from different entities and different countries. And so let me briefly introduce you to an amazing tropical cave in the very heart of the Yucatan Peninsula, which has been excavated and studied by paleontologists for over 2
Sorry,
decades.
Ella.
Yes?
Ella, sorry. I think we're not seeing your slides.
You are not seeing my slides?
No.
Oh, wow. Oh my God. So I have 20 minutes talking and you're not seeing anything? Oh, sorry about that. You see them now?
Yep.
Okay. If you don't mind, I will go very, very, very briefly to the previous. You see them there, right?
Yes. Yes.
Okay. Oh gosh. Very, very quickly. You see them there, right?
Yes.
Okay. Just wanted to show you the one with all the people that I mentioned are responsible for the work I'm going to be telling you, and the one about go right into the Loltún cave in the Yucatan Peninsula. The Loltún cave, Loltún means in Maya rock flower, flor de piedra, and it is a set of interconnected chambers with different units, chambers, tunnels, and the Huichil chamber with 2 units, El Toro and El Túnel, it's the places where most paleontology work has been performed. This Loltún cave has proven to be one of the most important sites for recovering quaternary tropical fossil remains, where around 68 species of fauna have been recorded from different soil strata. And there is a very detailed stratigraphic gradient described from El Túnel that encompasses nearly 17 layers that have been dated by different means. For example, layer 7 that has, based on horse bones, we know it's about 12,700 years old. And it has this stratigraphic gradient depicts a succession of from late Pleistocene to Holocene fossils and fossils, mainly from mammals, but which 11% of those are from rodents. But we also have even bones and fossils from tiny things like this little frog. Notably, these different stratigraphic layers of sediment that have been excavated they held different abundances of the fossil remains. And I have to, for example, regarding rodents, for which I have to highlight that most fossils are predominantly bone fragments that allow identification almost only to the genus level, generally only to the genus level. So these different abundances, for example, Sigmoron in the right, we can see that it was abundant throughout most of the Pleistocene and then significantly decreased afterwards. Whereas on the left, Eteromis became abundant exactly during the Holocene. And I'll want you to see these 2 panels. Orizomis and Ototilomis are the 2 species I'll be talking next. And the great majority of fossils have historically been found in temperate and in arid regions, whether recovered from ice permafrost, crystals, ambers, as bones, plant material, different things. In amber, we have found entire specimens. Consequently, most genetic studies done with fossils come from these temperate arid regions, from where DNA is currently really easy to obtain. In contrast, DNA is hard to preserve in tropical regions, especially due to high temperatures and extreme humidities. So, taking that into account, the Loltun cave fossils represented for us, the genetics and the paleontologists, an incredible challenge in terms of preservation of DNA and genetic genomic assessments. You remember the two rodents I highlighted? It happens that we had been working for some time with these two species, with extant populations of these two species. The first one, Ototilomis, is a genus with only two species distributed very concise distribution from the isthmus of Tehuantepec to the northern Costa Rica. And our phylogeographic analysis so far at the time had shown that it was constituted into three lineages, that is, three different evolutionary units, which the more recently diverged happened in the Yucatan Peninsula. At the same time, we were working with Oryzomis, which has a much wider distribution from northern United States along the two coasts, all the way to the northern Colombia. And this species exhibit a more complicated phylogeographic analysis, but it did also have a lineage that differentiated in the Yucatan Peninsula. And if we remember that from the Loltun cave, the paleontologists have excavated bones and hemimandibles from Loltun, we, taking all this into consideration, our own results and bones, we set forth to investigate several questions to evaluate, that we wanted to evaluate based on genetic approaches, which I'll tell you a little bit of in the following. But so the first concern we had is that if we could design a way to preserve the fossil samples, a means to preserve these tiny fossils. And why preserve? Because we had to destroy the samples we had, because we had to break them and use only where the teeth and the dentine were, which is a means where we could extract, most likely we could extract DNA. And yes, we went to our dear colleague, an optics physicist. He said this amazing photographic study for tiny fossils, and he developed an algorithm with which he was able to obtain high precision 3D images from both species, which became a novel optical technique for morphological digital preservation. So we solved that part, and we were able to go forth and destroy our fossils. And we had the following two aims, two questions. To confirm if this tropical cave was an actual reservoir of ancient DNA, in that allowed us also to confirm the identification of the fossils that were for the rodent species. And if we succeeded in this, we wanted next to build the first mitochondrial genome from these fossil mandibles of the two species. So for that, I'm not gonna detail you all the molecular and laboratory work that it meant, which was a lot done by Tania Gutierrez Garcia, just to tell you that we had 28 fossil mandibles from Ototilomis, 12 from Oryzomis, that encompass 6 different stratigraphic layers from 2,000 to 12,000 years old. We also use contemporary museum samples from specimens from museums. And first, we targeted a gene, only the cytochrome B gene from one species. And after the following part was to target the complete mitochondrial genome from the 2 species. And yeah, we successfully amplified ancient DNA from Loltun. For the cytochrome B, we have over almost 700 base pairs sequence, which even small sequence, but it showed high diversity in terms of different haplotypes, different sequences, both throughout and within the different stratigraphic layers. So based on that success, we followed the next analysis to amplify the mitogenome, the mitochondrial genome. We obtained different sizes from 2,000 to over 7,000 for the fossil samples, which is great success if you take into consideration the tiny fragment we had to work with. And for the museum samples, we have only almost the nearly complete mitochondrial genome. So we have the genetic data now. We could go next and ask so many questions. The first one was to evaluate if these ancient lineages, the fossil from the Holocene, were divergent from the modern ones, or if they were closely related. We didn't know what to expect exactly. And what we found is that for each species, the ancient individuals have their unique haplotypes, but in both cases, both species, these unique haplotypes are grouped with the modern ones. What that means is that despite the time, they are closely related to the modern ones. We also wanted to explore spatiotemporal distributions patterns, both our genetics and the paleontological ones, to discern a few or some of the Holocene biogeographic processes by contrasting the ancient and the contemporary lineages that we had. And for one analysis that we evaluated times of divergence, what we found was that the highest and latest diversification rates on both species happened concurrently in the Yucatan Peninsula, each with its own time and rate of diversification, but concurrently in the Yucatan Peninsula at the latest into the Miocene. We also found by evaluating temporal and spatial phylogeographic patterns, that means that the relationship among haplotypes, that both species are in agreement with different biogeographic features, which I don't have time to explain in detail, but for example, the Istmus of Tehuantepec, all of these acted as historical barriers, which reflects in the distribution of our different lineages that I've been telling you about. And coming back to the rodent, these rodent relative abundance patterns that I was telling you about, we can see that both of our species, Oryzomes and Ototilomes, are present along the stratigraphic gradient, but also that there is in here, for example, a recent period where Oryzomes, where the fossil remains of Oryzomes are much less abundant. And this allows us to support a biogeographic hypothesis where there is a localized change from humid forest, which is, by the way, the habitat that Oryzomes is more restricted to, which a period that was dominated by wet and colder climates, into another that changed in only a few thousand years to a warmer or drier climate dominated by grasslands, which is more closely to the habitat we see in this part of the Yucatan Peninsula nowadays. And with that, I want to thank you for your attention. Sorry for the mix of not having the slides up at the beginning, and I'm happy to take any questions you want.
Great, thanks, Eja. And we're gonna take advantage of our Q&A icon. So I'm only gonna read a couple of questions because we don't have that much time, but then we go back at the end of the session, then we can go back to the questions, okay? So the first question is from Gautam, and he or she, sorry, says that, do you think that there's any significant difference in fossils found across the different climatic biogeographic zones, like the tropics, subtropics, temperate, of the similar periods?
Oh, okay. If there are different
significant differences in fossils found across the different climatic regions.
Of course, yes, yes, no doubt about it. As I was saying, in the tropics, we do have fossils, of course, and they've been found in different places, even in Caribbean islands that are really wet and really warm throughout the history. But we have the temperate and cold climates are the perfect scenarios to preserve. You see, there's recent news that they found a complete bear with all its organs in the ice permafrost. So since those systems are better to preserve, we will have, the difference will be that we will have more abundances, more easy identifications of fossils, a bigger number of fossils that are preserved in all these temperate areas compared to the tropical ones. Loltun is a really gem because for reasons that are more paleontological, and it was able to preserve so many species and so many through so many periods of time. Yes, there are huge difference between temperate and tropical habitats.
Okay, so next question from Francisco Correa. He says, is it difficult for DNA to be preserved when global changes have occurred? However, it is possible to associate the divergence time of the groups with global changes and geomagnetic reversals? Are there studies of this type?
That is a complicated question first, because I don't know what is the last thing you said.
Possible to associate the divergence times of the groups with global changes and geomagnetic reversals.
Okay, I don't know what that is. I'm not a paleontologist or a geologist, but for the first part of the question, DNA, as I showed, we can estimate with very, very good certainty divergence times based on DNA once we have some calibration points to work with. And in this case, for example, we had plenty of calibration points because we knew the different ages of the different layers from where we had our fossils. So we work with DNA to date different events, whether they are from the fossils themselves, as in this case, or from contemporary samples that we can anyhow calibrate with fossils. So yes, and we could do that with climate change. Yes, we could. We can try to predict what will happen, or as based on what the presentation that Jorge showed us of the going back to the Chimborazo, we could use we could use those samples, the modern ones, and date the climate change.
Right. So another question, we have like 4 questions, but we're going to take them at the end of the session. But there's one that has been hanging there, which is from Olga Ricalde. She says that, have you done any comparisons with palynological data?
No, the answer is no. Our team is working with the zoological part of the excavations. There's people working with the flora. There's much less information and much less fossils from flora. It was harder in this cave to preserve pollen or that sort of thing that is common in other areas. No, so we are concentrating in the material that we are access to with more success possibilities.
Right. So, okay, as I said, we have at least like 4 or 5 more questions, but if you allow me, we're gonna take them at the end of the session. Please keep with us until the end of the session so we can move to the next presenter. Okay, thanks, Eja.
Thank you, Fabricio, and all.
Okay, so Leti.
Our next speaker is my present student, Ricardo Rivera Reyes, and he's currently finishing his master degree. I don't want to say the title wrong, so I'm gonna let Ricardo to tell you the title of his presentation.
Hello to everyone. Okay, my name is Ricardo Rivera. Oh, I need to share my. Okay, thank you, Leti, for introducing me. My name is Ricardo Rivera. I am a master's student, and now I want to tell you about my project, Elevational Patterns of Amphibians and Reptiles in Mexican mountain ranges. How is life distributed in the world and why is a central question by her. Especially the mountain, a bit of great interest in research. And many naturalists and scientists have researched this topic, such as Alexander von Humboldt. But why do mountains matter? Mountains harbor the highest diversity in many taxonomic groups, with about 25% of the Earth's lands covered by mountains. They are inhabited by about 85% of mammals, birds, and amphibian species. Today, at least 5 patterns have been described. Decreasing pattern, low plateau pattern, low plateau with mid-peak pattern, mid-peak pattern, and increasing pattern. The mid-peak pattern is the most common recovered pattern, and the crescent pattern is the second most common. Many explanatory hypotheses have been tested to explain these patterns, and we can classify into climatic, spatial, biotic, and historical hypotheses. Especially, climatic hypotheses have received more attention. Furthermore, we know the elevational patterns are affected by sampling for spatial scale and taxonomic group under study. Amphibians and reptiles are terrestrial vertebrates ectotherms, and they have received less attention on this topic than mammals and birds. But why do they matter? Reptiles are the most diverse group of terrestrial vertebrates, with more than 11,000 species known until today. And amphibians are facing a serious extinction crisis. Precipitation has been associated with amphibians diversity, while temperature with reptiles diversity. However, water energy dynamics, topographic heterogeneity, and its interaction with temperature and precipitation also have been associated with diversity patterns. Mexico has a great spatial heterogeneity due to large mountain ranges that cross it in different directions. More than a half of amphibians and reptile species are endemic to Mexico. And the majority of the species, including endemics, inhabit in mountain ranges, especially in south-central Mexico. What we did, we evaluated the elevational patterns of amphibians and reptiles in 5 mountain ranges of Mexico. Also, we test 6 models based on environmental variables as explanatory variables of observed patterns. Furthermore, we evaluate whether the spatial scale mediated by the amplitude of the elevational bands as grain size has effects on the observed patterns and their explanatory variables. We know that delimiting a mountain range is not always easy. And in Mexico, the mountain ranges are close to each other. So to do this, we use the Mexican phylogeographic provinces. Spatial records were obtained from the most complete Mexican pet fauna database. To start records and environmental variables, 14 transects of 54,200 kilometers were placed on the mountain ranges. Environmental variables were obtained for world climate and global aridity and pet database. The elevational data were obtained from digital elevation model of Mexico. Mountain ranges also have their own internal complexity. and there are environmental differences between slopes, mainly between coastal and continental slopes. So a gradient was defined for each slope with minimum 1,000 meters in elevation, at least 7 species recorded, and at least 70% of the length of the gradient with the spatial records. Also, the gradient with high sampling in 4. The sampling in 4 was quantified by the interpolation method of sampling coverage. The elevational ranges of species were delimited by their minimums and maximums records. Each gradient was sectioned in 100, 200, 300, and 500 meters elevational bands. Also, we calculated for each elevational band in each gradient the average of temperature, precipitation, and potential evapotranspiration, the standard deviation of elevation, and species richness. To evaluate the relationship between species richness and environmental variables, we use generalized linear models with a quadrant error distribution. When overdispersion was observed, a negative error distribution was used. Model selection was determined by corrected Akaike information criteria and value for small samples. We tested 6 models. Model 1 relates spatial richness to temperature. We expect more spatial richness with higher temperature at lower elevations, while at higher elevation, there will be less richness with lower temperature. Model 2 relates spatial richness to water availability, measured through precipitation. We expect more spatial richness with higher precipitation. Model 3, water energy dynamics, combines the effects of water and energy, assuming that the availability of water is limited by the amount of rainfall and by the amount of energy. So, high energy produces water loss through evapotranspiration, and low energy produces freezing. Then, we expect higher spatial richness where water availability is higher. Model 4 relates spatial richness with topographic heterogeneity, assuming that topographic heterogeneity generates a great number of habitats for the species. We expect more species richness with more topographic heterogeneity. Model 5 combines the effects of topographic heterogeneity and temperature. We expect more spatial richness with more topographic heterogeneity and more temperature. Finally, Model 6 combines the effects of topographic heterogeneity and precipitation. We expect more spatial richness with more topographic heterogeneity and more precipitation. What we found? More gradients were selected for reptiles, but more species records were obtained for amphibians. Total species richness was similar to the global trend, with more reptile species. The mean sampling coverage was high with values near to 1 in both taxonomic groups. The mean species richness per gradient was 35 for amphibians and 69 for reptiles. Our results confirm that mountains harbor the highest species richness in Mexico. At least 56% of the amphibians and 50% of the reptiles of Mexico are represented in mountain ranges. But these records might be higher because we only access near to 7% of the continental surface of Mexico. Furthermore, we know that sampling is heterogeneous and we had many information gaps. Principally, highlands need more sampling, especially at north of Mexico. What about the patterns? 5 patterns were recovered for amphibians. Mid-peak pattern in orange was the most common pattern, with more relevance in continental gradients. The crescent pattern in red was the second most common pattern, and principally in coastal gradients. More types of patterns were recovered in continental gradients. For reptiles, 4 patterns were recovered. Again, mid-peak was the most common pattern, especially in continental gradients. Again, decreasing was the second most common pattern, and again, with more relevance in coastal gradients. In this case, all non-intermediate gradients were in continental gradients. Our results are similar to Rabeck and Gao, who reported that mid-peak is the most common pattern in elevational gradients. At least 55% of the 55% of the gradients show the same pattern across a spatial scale. But in some patterns, spatial scale affected in 2 ways. First, increasing in the spatial scale may appear to change the elevation ranges of species. And the second, increasing in the spatial scale produce change on the shape of the pattern. Here I show you the same pattern at different elevational band scales. And we can see how it changed from mid-peak pattern at lower scales to a low plateau with mid-peak pattern on larger scale. Pattern shape was more frequently modified at low elevations. Explanatory variables of biodiversity. For amphibians, only 5 of the 6 models were selected. Only model 6, interaction between topography, heterogeneity, and precipitation was not selected. Model 3, water energy dynamics, was the most selected at the smaller spatial scale with high fit and with more relevance in continental gradients. Okay. Model 1 and Model 4, temperature and topography heterogeneity, increasing their selection with the spatial scale. But there is more relevance of temperature for coastal gradients and topographic heterogeneity for continental gradients. Model 2, precipitation, was only selected in continental gradients with low to medium fit. For reptiles, 6 models were selected. Again, Model 3 was the most selected at the smaller spatial scales with high fit and with more relevance in continental gradients. Okay. Again, Model 1 and Model 4 increasing their selection with the spatial scale. But in this case, the temperature had more relevance in coastal gradients compared with the amphibians. Model 2 was the only selected at largest spatial scales with low to high fit. Only continental gradients selected all models at the largest spatial scales. But what is the process behind the selection of the models? Okay. Maybe the first thing we ask yourself was, why precipitation was not selected? If it is being associated with herpetofauna richness. We think it is because, on one hand, in mountains, water tends to flow away from where it falls. And on the other hand, water loss for evapotranspiration is not constant. So we have places with more evapotranspiration than others at the same elevation. In the coastal gradients, humid air from the ocean generates fog. Oh, creo que perdí la señal. Can you hear me?
Yes, yes, we hear you, Ricardo. Yeah.
Oh, sorry. Okay, I am here. Okay, water loss for evapotranspiration is not constant. So we have places with more evapotranspiration than others at the same elevation. In coastal gradients, humid air from the ocean generates fog that spreads along extensive areas in mountain ranges. This fog represents an important quantity of water that condenses on the vegetation, and this water is not accounted for in precipitation measures. For this reason, precipitation does not necessarily reflect an adequate representation of available water. That is the reason for the Model 2 being unsuccessful to explain the spatial regions patterns for either reptiles and amphibians. Therefore, in places where the rain and the fog exist above transpiration, energy becomes more relevant for species distribution because water is not limited. Thus, that is the reason for that the model one explains the spatial regions patterns better along coastal gradients. Now, in continental gradients, in places where the water is limiting factor, the water energy dynamics become more relevant to predict the spatial regions pattern. On the other hand, topographic heterogeneity is a better predictor of spatial regions at coarse scales. We think this is because such heterogeneity is better preserved at larger scales compared to small spatial scales. It's more likely to capture a wider elevation range if you have a larger area. Finally, the interaction between topographic heterogeneity with temperature and precipitation seems to have relevance under more particular conditions, but with our data, we could not detect any tendency. That may be the reason that Model 5 and Model 6 were less selected. Conclusion, the Mexican mountain ranges are an important reservoir of amphibians and reptile diversity. The distribution patterns were similar to those of other taxonomic groups at different spatial scales. Explanatory variables showed a great complexity between the grades and across the spatial scales, especially in reptiles. This is of great relevance for its conservation because the factors that affect biodiversity vary in the same way between mountain ranges and across spatial scales. So, many thanks to CONACYT, to Posgrado Ciencias Biológicas of UNAM, to my tutor, Leticia Ochoa, to the organizers, and all the people behind the International Humboldt Day, to all of you for staying here. Thank you so much.
Thank you, Ricardo. We are going to start with questions. The first one is kind of difficult. I actually don't know the question, the answer, but let's. So it's from Damián Villaseñor Amador, and he asks you, what was the herpetofauna that Humboldt saw in Mexican mountains?
I don't know.
I don't know either.
Only I know that Humboldt studied the vegetation, especially in the Cofre de Perote. It's my knowledge, okay? But I don't know if Humboldt studied herpetofauna. I don't, but I can
Find out.
I can find out more, maybe.
There is another question from this. Actually, there were two questions. Also, would it be better to use reptiles and amphibians to model biodiversity of a mountain range rather than mammals or birds because they are more sensitive to temperature or rain changes? I think he meant in the global change scenario.
In the case of the reptiles and amphibians, we think that they are more susceptible for environmental variables. But the truth is, we have more or less information about them than mammals and reptiles. But mammals and birds also have showed sensibility with environmental variables. In the case of the bats, precipitation also have been associated with their elevational patterns. But we need to evaluate more than amphibians and reptiles. If I can explain me well, please grade me or send me a mail. And in writing, I can explain more better. Sorry.
Don't worry. Francisco Correa asks you if you think that geomagnetic reversals that this, I'm going to read it fully.
Mhm.
Throughout the history of Earth, there have been changes in the polarity of Earth. So it means that the north, magnetic north changes. In these geomagnetic reversals, the changes that occur in geographic patterns being determined, I mean,
I don't
think the
know.
question is if they can change.
I don't know. I don't read anything about this relation in the magnetic and geometric reversal magnetic. I don't know, but sounds very interesting. If you have any paper, please send me to read about this item, please.
And for now, the last question, we will do more at the end, is from, sorry, the last question was from Francisco Correa. Apologies if I didn't say the name. Is from Silvia Torres. And she asked you if you know which reptiles or which species of reptiles are currently the most affected by global warming.
Okay. Especially by under global warming, I not sure, but in the global axis of reptiles, the turtles and some families of lizards. I'm not sure what families of lizards, but turtles is the most endangered group in reptiles.
Thank you, Ricardo.
No, thank you.
Fabricio, I'm going to share, give you the mic to present the last of our panelists. Please stay tuned.
Yes, do so. Please stay tuned until the end, so we can have we can take your questions and all the comments that you have mentioned. We are going to read it at the end, and we can actually even give you some audio time or video time, so you can ask directly to our panelists. So now for our last talk, I have the pleasure to introduce one of our PhD students here at the Instituto de Ecología in Mexico, which is Pedro Luna. Pedro is a senior PhD student here at INECOL, and he did his master's also here at INECOL, and he came from the University of Puebla, where he did his bachelor's. He's mostly interested in interaction networks with invertebrates, but he's now taking a more biogeographical, macroecological perspective, and he's trying to expand the scope of his studies that he's more used to conducting in local scales, but now he's moving more to large scales and geographical scales. And the talk that he's going to present today is going to show exactly that. So with that, please, Pedro, if you could unmute your mic and share your screen, and please.
Hello, can you hear me?
Yes.
Okay. Hello to everyone. It's a pleasure to be today here and an honor for me to participate in this kind of event. Well, I'm going to start talking about my PhD project. And in the context of this event, the title is a Humboldtian view of species interactions across elevational gradients. I am Pedro Luna, and these are my advisors and my collaborators. Okay, well, in 1802, Alexander von Humboldt went up a mountain and came down with an idea that changed ecology. This idea is that environmental conditions influence how biodiversity is distributed over the Earth. In a fast example, we can say that the species richness increases in lowlands, in the mountains, because of the increases of temperature. This idea changed the way we study biodiversity. Nowadays, the study of environmental conditions shaping biodiversity is a cornerstone in ecology and remains as a widespread heuristic approach. One of the most recognized relationships between environmental conditions and diversity is the relationship between latitude and species richness. As you can see in this map, the tropics, well, we can see that red colors denote higher species richness. So we can see that in the tropics, we have higher biodiversity or higher species richness. And this can be explained, for example, by temperature or by primary productivity. However, to fully understand how biodiversity is distributed, we must consider elevational gradients because mountains harbor the majority of Earth's biodiversity, but in the tropics. As we can see in the map, we have the Mexican Transition Zone, the Andes, the Great Rift Valley, the Himalayas as biodiversity hotspots, and they are also elevational gradients. However, if we see another elevational gradients out of the tropic, the Alps, we can see that this hotspot of biodiversity is not as high as in the tropics. So we have to acknowledge that the location of the mountain or the location of the elevational gradient also affects how biodiversity is distributed. And why? Depending on where the mountain or where the elevation gradient is located, there are different changes in the environment. For example, in the tropics, as we go up into a mountain, the changes in temperature are very drastic. For example, in lowlands, we can have a jungle, and if we go up, then we can find a cloud forest. And if we go up, we can find temperate forest. This only because of drastic changes that occur in temperature. But if we go to a temperate elevational gradient, these changes in temperature are not as drastic. So the environment is more homogeneous. Okay, we have to consider also that environmental conditions are not only influencing species diversity, because diversity is more than just the number of species. Biodiversity has many components as genetic, functional, taxonomic, and interaction diversity. Therefore, species interaction, in this case, could change with environmental conditions. Assemblages change over nature, turnover of species, for example. Their interactions are also expected to vary because of these changes in species assemblages. Under this approach, using ecological networks, we are visualizing communities as ecological networks in which we have species denoted here as nodes and interactions denoted as links between species. And well, this species interaction, these communities interacting can change across temporal gradients, for example, across different seasons, or across special gradients. In this case, we can talk about an elevational gradient. The study of species interactions is part of Humboldt's view that we live in a world where everything is connected and in which environment shapes its biodiversity. As once he said, the discovery of a new genus seemed to me far less interesting than an observation of the geographical relationships of plants. This is something that Humboldt wrote, and it's a very interesting idea of how everything is connected and how we should explore this connection among living organisms. Well, ecologists have mainly studied these patterns of how species interactions vary over space by studying how trophic specialization is structured over space. By trophic specialization, I mean to the use of resources by species. This is niche breadth and overlap. However, there is not consensus over such patterns, over how trophic specialization vary over space. There are several evidence that have shown that species interactions become less specialized in the tropics and become highly specialized towards temperate regions. And there is evidence that this could be exactly the opposite. Generalization or less specialization can occur outside the tropics, and specialization is a characteristic of tropical regions. One of the most interesting part of these patterns is that temperature, climatic stability, and plant richness can explain these gradients of species interactions. This has been studied with floral plant and floral visitor networks and with frugivorous networks. Um, However, we have also to consider that trophic specialization also vary over elevational gradients. Just as species diversity, elevation becomes fundamental to understand how and why species interactions vary. So we can expect to find changes in how species interact from the bottom of a mountain to its top. And this could be because a change in temperature, for example, as it has been shown with floral and plant interactions, floral visitor and plant interactions, and for frugivorous networks also. Following these ideas, we aim to assess how trophic specialization varies over elevation under two approaches, a global approach and a local approach. For the global approach, we assess how trophic specialization varies over elevation, taking into account the effect of environment over tropical and non-tropical elevational gradients. These characteristics that I explained to you about how environment change over different kinds of elevational gradients. For this approach, we use 87 networks of plant floral visitors that were compiled from databases in internet. And we also used geographic information systems and several bioclimatic rasters, temperature and net primary productivity, to see how trophic specialization is structured over space. Here we have a map of these 87 networks, and we found a total of 46 networks in the tropics, denoted by the green color, and 41 networks outside the tropics, denoted by the blue color. The size of these balls, these bubbles, denote the number of networks that were found by us in different places. Bigger bubbles are more networks. Was similar in order to make further comparisons, and we found that yes, indeed, the elevational range of these networks is similar. And for these 87 networks, we measured a very known index for ecological networks, that is the complementary specialization index. This index ranges from 0 to 1. In 0 values, it denotes low specialization, highly connected networks with a lot of niche overlap. And in 1, this index denotes high specialization, low overlap, and not very connected networks. One characteristic of this index is that we use weighted matrices. This is the interaction frequency between species, which is a more straightforward approach to measure specialization. Well, what do we found? We found, according to previous evidence, that trophic specialization is lower in the tropics, just as some works that have already reported these trends. We see that the tropics harbor less specialized networks in comparison with the non-tropics. However, when we go into the elevational gradient, we can see that there are changes as elevation increases. One of the most important trends that we found is that trophic specialization decreases towards lowlands in the tropics, as we can see in this graph, elevation and complementary specialization. And this green line denotes how specialization decreases with elevation only in tropical regions. We did not find any relationship in non-tropical gradients. And well, we think that the lack of structure in non-tropical elevational gradients could be a result of the less environmental variation that occurs along these elevational gradients. And well, here we can see a map of how this is distributed. Here we have the index Red colors denote higher specialization, light colors denotes lower specialization. And if you can see, this pattern is quite clear. There are a lot of red, orange dots outside the tropics, and there are more light dots within the tropics. Of course, we can see also the elevation of these places. And we also can see that in the tropics, there are big dots which denote higher elevations with less generalized interactions. But what about environment? Well, trophic specialization decreases with higher temperatures. We can see mean annual temperature and complementary specialization. And we can see that warmer places harbor less specialized interactions. This could be the case, for example, of the tropics, which are warm. And there is also one explanation to this, that also temperature explains higher species richness. So why we are observing this? Well, this could be because in places with higher species richness, we have higher functional redundancy. A lot of species doing the same functional role. So higher functional redundancy leads to highly connected networks. This means less specialized networks. In the case of net primary productivity, we saw that trophic specialization increases with primary productivity. Places where plants thrive, where there are a lot of plant-derived resources for animals, interactions become more specialized. This could be related also to foraging, as species, as in a place where there is a high net primary productivity, we can have also higher species richness, which could allow to animals to search for preferred resources, which in turn could lead to higher specialization. Well, we can explain the specialization of species interactions over latitude and elevation because of the change of environmental conditions over can be found in places where species diversity might be higher, regardless of elevation or latitude. This is that environment is more a more straightforward explanation than just space. Specialized and less connected networks can be found where plants thrive, places where the forage of preferred resources should be favored. But this was done at a global scale. What's going on at a more fine-grained scale, or if we can find these same results in a single elevational gradient? Well, for this, we are going now to the local approach, in which I studied plant flower visitor networks over a mountain in the Mexican transition zone, which is located in this place, as is shown in this map. And this mountain is the Cofre of Perote, Cofre de Perote, which is 4,280 meters above sea level in elevation. And these are preliminary results of one of my chapters. Well, first of all, we have to say that Humboldt visited this mountain. When Humboldt visited Mexico, he entered from Acapulco in the Pacific Ocean. He traveled around the center of Mexico, and he left Mexico from Veracruz port, and he visited this mountain, which is located here in the red circle, the Perote Mountain, which is very near where I live and where I'm studying in the Ecole Institute of Ecology. This mountain is 4,292 meters above sea level, and the tree line of this mountain reaches up to 3,700 meters. Okay, across this gradient, we set 11 sampling points with a difference among them of 500 meters between them. On this map, we can see all the sampling points, starting from the coast, going up to the mountain. Okay, so along this gradient, we have a lot of environmental variation. We have 0-meter coastal dunes, which are very hot. As we go up, we can find tropical dry forest at 1,000 meters above sea level. At 2,000 meters, we can find cloud forest, we can find rainfalls, and very exotic forests. And near to the top of the mountain, we can find temperate forest like this Avis religiosa forest. Along all these places, I sampled all the possible interactions that I could record of plants and their floral visitors like insects or other arthropods. It's important to note that along this gradient, temperature changes and decreases toward higher elevations. The coastal dunes, we have almost 30 degrees Celsius, and in the 3,500 meters, we can have 10 degrees Celsius in temperature. So these changes are very, very drastic and expected, like in every elevational gradient of the world. So what do we found? Well, first of all, we found that this graph shows the number of plant species that are visited by arthropods. So if this 20 here denotes that in the coastal dunes, we have 20 plant species that are visited by several insects and arthropods. But we found a peak in this trend. We found that within the cloud forest, there are a lot of plants that are visited by several arthropods, with the highest number of plants being visited at 2,000 meters, which is the cloud forest. The plants that were more visited by insects and arthropods were the family Asteraceae. This family was highly visited, also is highly abundant. The next family was Fabaceae, then Lamiaceae, and finally Malvaceae. In total, I found 37 families of plants being visited by arthropods and insects along this gradient. And well, but we are talking about elevation. So as these are preliminary results, I'm going to show you these results. This network here is a network from coastal dunes at 0 meters above sea level. And this network here is from 3,000 meters, almost in the top of the mountain. The complementary index, specialization index, showed that in the coastal dunes, interactions are specialized. And in the pine oak forest, 3,000 meters, interactions become less specialized. As you can see, even the network is highly connected. And in this part is not as connected as this one. What's going on? What can we say about these results? Well, I suggest that this change could be because of the change in environmental conditions. As I showed, that the temperature decreases as elevation increase in this gradient. So we still don't have all the results, but this is the hypothesis of the work. And we hope that this trend we hope that we will report this trend in the future. And as you can see, this is also similar as the global results. As in the global results, we found that in low elevations, interactions are more specialized, and in high elevations, interactions are more generalized or less specialized. So to end this talk, my take-home message is that the variation of environmental conditions is only a set of factors that explain how species interact. Environmental conditions are not the only factors that explains the variation in interactions, but this is one factor that could explain these changes in how species interact. And we can also say that the way species use resources change from the tropics to temperate regions, and from the sea to the top of a mountain. And studying species interactions and this relationship with environment is an Humboldtian view of species interactions across elevational gradients, something that not many people has talked about it. Is now like we can study species interactions with environmental conditions, but this idea came from Humboldt and his expeditions. And this is why this talk is entitled like this. Thank you very much. I appreciate the invitation to this event, and I have to say thank you to all the people who helped me in the field. And this is my mail and my website if you want more details about this research. Thank you.
Excellent. Thanks, Pedro. Thanks for the great talk. And we have some questions before inviting all the panelists back. So I'm only going to read a couple of questions, 2 or 3 questions. So from Andrea Raz, she says, you say that biodiversity in the tropics is greatest on mountains, right? But does this apply only to terrestrial environments? What about coral reefs?
Mm-hmm. Well, that's a very nice question. Well, I don't know much about marine environments, but I know that there is a gradient that goes from coastal lines into the depth of the sea. And yes, there is higher diversity in coral reefs, but this is the contrary of elevational gradients in the earth over sea level. Higher species richness in coral reefs, and then it decreases towards deep sea.
Right. And then from Professor Elva Escobar, she asks, do these interactions and connectivity patterns can be observed in freshwater or saline lakes as well?
Well, I have never seen any of these kind of studies in freshwater environments or in lakes. There are people studying, for example, freshwater fishes patterns in the world, but I don't know if they have interaction data. This kind of data is hard to obtain. I know that there are some studies with amphibians, which are studied on fishes, for example. And yes, if you can have 2 species and determine how they interact, you can apply this approach.
Right. And also Elva says, just to complete, why is water availability not considered in your gradient?
Well, we consider water, but these results are not shown here. We consider precipitation and precipitation stability for this, and there is something about mainly precipitation has something to do with the flight of insects, for example. Precipitation also affects the pollen of plants and dilutes nectar. So yes, water is has something to do with this, not only temperature and primary productivity.
Great. So last question, and then we can invite back all the panelists, and then we can have a discussion around the questions that are already there. So last one from Damián Villaseñor Amador. Will interactions become less specialized, more generalist, as global warming increases and the species move upwards in elevational gradients?
I don't know, but yes, generalists are the winners in this game, because they are very dynamic. There is one study in Europe that found that specialization is not explained by environmental changes, but specialization is explained by functional traits. So if you have the traits, you can interact. So generalists have almost have dynamic traits. For example, they can interact with a lot of species. So I think that generalists could be good players for climate change.
All right. So with that, we thank Pedro, but we still have some questions for Pedro, but then we can ask them now in our discussion session. So I invite back Leti, Ella, Jorge, and Ricardo. and we can read some of the questions that are still unanswered. So if they are specific, we can say that, and if not, we can just put it out there, and anybody can answer if you agree.
Fabricio, do you think it will be okay if I ask the first question, because it's a very particular question, and then we can go back to the more ecological thing.
All right.
So Emilio Jorge Carrera Mendiola asks, this question is for Jorge. Do you think, Jorge, that without wanted Humboldt incentive or promoted the invasion of the US to Mexico when he shared the maps that copied from the Colegio de Minería? When he went back to Germany, he went through the States. And do you think that would be possible? That could have been possible. Oh my God.
Well, I don't really think so. That's a very strange idea for several reasons. Humboldt left Mexico in, I think it was 1803.
1804.
1804? No, it was 1803, no, because he spent just 1 year in Mexico.
It was from March 1803 to March 1804.
Okay, so I was 1 year wrong. And the US invaded Mexico, and that was still a Spanish colony. In the analysis of the socio-economic structure of the colony, Humboldt already perceived that there were some indications that Spain would probably lose these colonies shortly, because there were many signs of that. And he was very, very clever to identify those signs. But I mean, there was no country called Mexico at the time. Sometimes we who live in Mexico have a hard time imagining a world without a country called Mexico in it, but there was no Mexico as a country. So this colony, New Spain, was very big and very difficult, very heterogeneous, and probably it was not well structured because of the large territory and the loose connections between the different parts. The other important thing, and then the US invasion to Mexico took place after Mexico had become independent,
Yes.
probably half a century later. And I think Humboldt was a very old person at the time. The other thing that must be stressed also, that many of the maps that were available at the time for Mexico were wrong. And Humboldt did a lot of work correcting those maps. It's really interesting to read his description of the moment when they were supposed to arrive in Acapulco. And he had the map in his hand, he was saying, Acapulco is not here. I mean, the maps were so wrong, and he was correcting it with all his instruments, maps all the time. So I cannot imagine that the maps that he copied from the College of Mining in Mexico were so good maps. Obviously, he knew a lot about the potential resources that Mexico had. Some people have claimed that he was a spy of the German government sent to the Spanish colonies. At the time, all these countries were not countries, but Spanish colonies. There has been a lot of discussion of that, but just to think that the sharing of these maps with the US government by Humboldt, if he did, promoted the invasion of the country 50 years later, it's a little bit strange. I don't think that's the way things happen, but that's my opinion.
Okay, so let's move to the more ecological biogeographical questions. Go ahead, Fabricio.
Right. So this one, for example, can be answered by anybody. If you guys wanna answer this one, this Amaury Diaz Solis asks us, Humboldt and his colleagues did some work in arid zones or semi-arid zones. Are you aware of any of those works of Humboldt in dry areas?
Well, he certainly visited Colombia's coast in the arid region.
Mhm.
The story of the arrival of Humboldt to the Americas is quite interesting because he actually took a ship that was carrying them to Mexico. His intention was to visit Mexico, and I think that was a disaster for Mexican biodiversity and biogeographical studies because there was a disease outbreak on this boat, and they have to land in Venezuela, well, in what is today Venezuela, as an emergency landing because the captain didn't want to continue with this trip. And they were left there on this little town, I think it's called Cumana. And then he turned around and saw the absolutely fantastic biodiversity of those latitudes and spent many years there. And a consequence of that is that Mexico was not really visited from the biological point of view as profoundly, as carefully as he did in Colombia, Ecuador, and Venezuela. So that was an accident of history. And he went to some relatively dry areas, but more temperate in Mexico. He traveled from Acapulco to Mexico City, so he definitely went through the Balsas River Basin, which is dominated by tropical dry forests. But I don't think he spent any significant time in deserts or in desert vegetation, although some of the Andean plateaus that he visited were quite dry.
Right. Thanks, Jorge. There's one related to that, and from Susana Alvarado. She asks, was the idea that land use change modifications impact climate already around Humboldt's time? Did he write anything about it?
He was pretty much aware of that. Yeah, I don't think that he conceived that as land use change, but definitely he already noted. I mean, the conceptual framework at the time was completely different from ours. But yeah, he already noted that there were some special areas where human activities were easy to conduct and other areas where they were more complicated. And that was having an impact on the distribution of plant species and animal species. He noticed that mostly on the Andes, because before in the rainforest areas that he visited in Colombia and Venezuela, for example, when they went through the Casiquiare River that connects the Orinoco with the Amazon basin, that was almost completely untouched. Human settlements were very small. But then by the time he arrived in the time he spent in Bogota and all the Andean highlands, he was aware of the human impact that was already affecting those areas at the time. We cannot hear you, Fabricio.
Oops, sorry. Okay. Anybody else wants to fill in on that question? Asia, you have some idea of that, or?
No. What Jorge said is it's what takes it.
Excellent. So this one is answered, and then we have another more specific ones, don't we, Leti?
Yes, but before that, Mario Rubio, I lost the comment, but he said that he did that. Humboldt did visited Miguel Rubio did visited dry areas like the llanos in Colombia. I don't think llanos are really dry.
Llanos are not dry areas, no.
Not desertic areas.
Exactly.
Those are the savanna regions, the large savanna regions of South America, yeah. They are seasonal though.
Very seasonal, yes.
Mm-hmm.
Okay, so let's do a specific question. Should we start in order?
Yeah.
Yes, let's do that. So Andrea Martinez asks, this is a question for Ella. Ella, have you tried 3D printing for the preservation of fossils? Or would you rather keep it only as digital information in a database?
Okay. We certainly want to just keep it in digital for many reasons. One is that if you go into the anthropology zoological museum where these Loltun fossils are kept, Loltun and all of Mexico's zoological, archaeological remains, you just can't put one fossil more in there. It's like full rooms full of fossil remains. So of course the digital is the best option. Nonetheless, we could certainly, from the 3D algorithm that Dr. Gutierrez developed, we could easily print a 3D fossil or a 3D but no, we don't have a space. We're even there's a plan to digitalize some more fossils and probably not keep all of them, every single piece on these places museum, because there's just not enough space anymore.
Great. There's also, so let's go for another question for a different panelist. I think, okay, for Pedro also, I lost some questions here. Did people, yeah. Anyhow, Elva, just one comment. Elva says that there's a lot of publications of limnology that do not only consider fishes, but also invertebrates, that she invites you to take a look at it. And the other one is from Francisco Correa. Could trophic specialization be seen as a result of selective pressure against extinction?
Okay, let me think. Trophic specialization be seen as a result of selective pressure. Well, under a network approach, specialization is measured at a community level, first of all. And well, of course, evolutionary and speciation dynamics have something to do with specialization. But I don't know if extinction is one of these factors. For example, in this approach, if one species interact only with another species, this is not considered as specialization. This is only two rare species interacting. So species that are in risk of extinction are not the species that are more specialized, for example. This is, well, my answer for this.
Right, right.
Well, and also the rare species are the ones that are in the higher risk of extinction. No? This is why.
Okay, thanks.
Yes, so this is a question for all panelists. Juan Hector Garcia Chavez asked if, comparatively, how is the, I forgot the word. How could you see the contribution, yes, sorry, the contribution from Humboldt and comparatively to the expeditions of Mocinho and José Mocinho together, José Mariano Mocinho and Martín de Cecé. Do you think it's possible to compare them? Anyone?
I wouldn't know how to compare them. They had different aims, and I think they had different trainings and different purposes. I mean, all of those are really, from the historic point of view, really important events and amazing contributions of people who were really promoting the study of nature as it had never done before. But I think that any comparison would not be completely fair. I wouldn't dare to say, oh, that one is better than the other, or that one had a more profound impact. However, Having said that, I would like to mention the fact that Humboldt had a very universal thinking in scope. For example, even though he was a very proud Prussian citizen, there was no Germany at the time, it was Prussia. He had no problem publishing his results and his papers in French because that was the dominating language in science at the time. And I don't think that he ever suffered from the fact that, oh, I'm affecting my language because I have to publish in another language, because he was aware of the fact that if you would publish in French in those years, you would reach a much larger readership than writing in German, that is such a complicated language. And I think that so many people were able to get familiar with his works. Even Darwin, for example, it's amazing to see, if you read carefully The Origin of Species, you can feel amazingly how much Darwin read Humboldt. And I'm not sure that Darwin would have been able to read in German. Of course, Humboldt's works were later translated into many languages, into Spanish, I think only in this century, which is interesting. But I think that we really have to conceive science as a universal phenomenon and try to break barriers as much as possible, and not to try to impose those barriers through language. English is the language that we speak now in science, and it won't be like that forever, and it hasn't been like that forever. But it's the only way we really have to share what we think and what we learn with our colleagues from all over the world. I insist every time I speak with my students that I have learned so much from my colleagues in Vietnam who study ecological systems that are very similar to ours. And the only way I can learn about their work is because they publish and speak in English to me. And I think that's something that Humboldt also promoted a lot, and that's why he wrote most of his work in French.
Leti and Fabricio, there is one person that raised hand to do a live question. Giannici Ottaviani.
Oh yeah, we can, okay.
Hello.
Talking is permitted now. Yes, please, Gianluigi.
Thank you very much for the beautiful series of talk, very inspiring. And I was actually fascinated, particularly about this amazing gradient that you have in Cofre de Perote from the ocean. And actually, I think it's an amazing experience, a natural experiment for testing some, community ecology, biogeographic questions. And actually, linking also to one of the series that we had yesterday about sea islands, sky islands, and islands on the sea. Since some of the expectations from Pedro Luna was about the loss of specialization of interaction network in higher elevation. So would that be also, are you also thinking along those lines of linking to the island biogeography principles and concepts in this regard, on this regard?
Okay, well, the relationship between, well, one of the topics that we have explored with species interaction is the species area relationships. If there is the question is if there is a species area relationship or a interactions area relationship. We have some studies about this, and we might say that there is a relationship between area and interactions, but in this study, we did not take into account the area. We are taking, well, we could take into account the area, but we sampled the same equal plots all around the gradients. So for this data, it's not easy to test these assumptions, but for other systems, it could apply. Also regarding the movement of species, also in the Island biogeography, I could think of doing some analysis with some special tools, but this could take a lot of time as it's not part of my thesis, but I have been thinking of in this too.
Yeah, it's very interesting indeed, because yeah, definitely the relationship with the area, it's relevant, but also what defines insularities indeed is also a combination of other parameters such as temporal and spatial isolation. So I think that this would be a nice addition, because you will have this cross-taxon interaction between plants and animals. It's really and along this huge gradient within, having different biomes in a matter of just 100 kilometers or less. I mean, it's super fascinating. So I'm very looking forward to see results coming out from you guys. And thank you again for the great talks.
Thanks. Actually, I would like to take the advantage of this comment by Jean-Louis and the question to share this phrase from Humboldt. If you guys are looking at it now. So when Humboldt came through Mexico, so he entered in Acapulco and went to Mexico City and then Guanajuato and certain mine areas, as Jorge was telling us, here in Mexico. And then he left Mexico from Veracruz, from the port of Veracruz. So he said this in the political essay of the Kingdom of New Spain. So in one day, the inhabitants of Veracruz, which is in the Gulf of Mexico, descend from the area of perpetual snows to the plains, immediately to the sea, where there is a heat that suffocates. Nowhere is it better to see the admirable order with which the different plant associations are happening, one above the other, that when one goes up from Veracruz to the plateau of Perote. So that in this wonderful country, in the space of a few hours, the man of science travels the full scale of vegetation. So we can actually discuss around this idea. It was actually our inspiration with Leticia and I about making this symposium and inviting these speakers under this Humboldtian view of that everything is connected, and being the first ecologist of modern times, we were inspired to invite these speakers, and maybe we can discuss a little bit more about this, the influence of this Humboldtian view in our Mexican biogeography and our Mexican research. If any of you guys want to comment on that.
Well, yeah, maybe just a quick comment. I have had the opportunity to travel from Mexico City to Veracruz with many colleagues from abroad for different purposes, going to Los Tuxtlas, for example, or going to Oaxaca. And that's a trip that we take for granted. Now we just go down from the highlands to the lowlands. And I mean, that's something that we have done many times, those of us who live in central Mexico. And it's really amazing to see their faces, particularly when you're coming up, for example, from Veracruz, and you're able to see the Pico de Orizaba, which is the tallest mountain in Mexico. Also almost 6,000 meters, a little less, and well, 5,600, something like that. And the continuity of this gradient in these mountains that are so close to the sea, it's amazing because, for example, you go to the Himalayas and the elevation gradient is much wider, but the sea is much farther away there, you know? And so, yeah, in many places in Mexico, you have these very, very steep gradients. And I think there are many opportunities, for example, also in the southern Sierra Madre, in Sierra Madre del Sur, in Oaxaca, where I have worked with one of my PhD students. There are some places where you have continuous gradients with vegetation and ecosystems in relatively good state still, although that's changing very rapidly, all the way from sea level to places that are almost 3,000 meters elevation. And maybe we can take advantage of those places to conduct research like that described by Pedro in these places that are so interesting and we have already so much information, but still we are lacking understanding of what's happening really there.
Right. Thanks. Eja?
Yeah, I would probably in also in the aiming of wrapping this up, I will insist that we should also think of the legacy of Humboldt, not only for Mexico, but for biogeography in general, is those other gradients that we have in nature. Of course, the elevational, the latitudinal ones are probably more graspable for us, but there is the depth. There's a depth one in freshwater lakes and in oceans, and also these historical ones. All this historical information that we can obtain from stratigraphic gradients or other kind of historical gradients are necessary information for us to understand what we are seeing in biodiversity distribution now in the current times, and also to be able to think of ahead to the future. And I hope I was able to show that as neatly as Humboldt and Pedro and Ricardo and Jorge and all of you can describe the climate and the vegetation associated changes in these gradients that we see in mountains and latitude, we do see in stratigraphy. And these abundances that I showed are tightly associated with climatic changes and the vegetation changes associated. So there are many gradients to study and to work with.
Excellent, excellent. So we have some questions that we're going to give them to the panelists, which are very specific questions. We have a couple for Pedro and I think one from Ricardo. And just a comment here from Francisco Correa. It says, just as biodiversity in Mexico impressed Humboldt, it continues to impress young biologists, of course. And I think it has been the engine or promoter of biodiversity studies in Mexico. Of course, we have forgotten about some of the things, but actually that's one point that I would like you to comment about. It's are we actually getting that inspiration, or just until we get into the biological studies, like professional studies, that we can actually get that inspiration? So are we capturing that inspiration in younger students or not? That vision or that motivation for the studies? What do you guys think about?
I would like to say something about exactly that. That
Please.
is the speciality of Jorge. I don't know how to express it, but when you are young and impressionable, and you have a teacher just like Jorge, and this idea that he's trying to impress, that everything is connected with everything, is not from him. Humboldt had already taught it, but that everything is connected with everything. That you need to understand magnetic fields, climate, evolutionary processes, mountains, paleontology. You need to understand a lot of that to actually try to answer a very, very specific question in a site, in your thesis, in your project. I'm sorry, Jorge, to say it again, but it's true. It's that maybe what we need to impress that in younger people, to actually make them see that everything is connected with everything, and I think that's the beauty of biogeography.
Exactly, Jorge.
Yeah, thanks a lot, Leticia. I'm really impressed. I didn't know that I had such an impact on you. I'm glad it was a good impact. Could have been the other way around also. I also think that it's important that more people involved in research, who are doing actively in research, get more involved in teaching at lower levels. I think that a lot of undergraduates would benefit from hearing to really impacting lessons from many people who are only working at institutes and normally do not participate much in undergraduate teaching. I think we are sort of abandoning undergraduate teaching very much. I mean, I'm not, because I work at the faculty and I have to deal with hundreds of students, but I've noticed that so many people who would have so much to say to younger students are not interacting with them. And I think that's really a pity. And the other thing, I really think that we should encourage young students to read Humboldt's works. All of them are published now, are available. Well, maybe not all of them. I'm not totally sure. But many of them are already translated into Spanish. And I think many people read about Humboldt. There are so many books. For example, I was seeing here in the comments about this, the Invention of Nature book, which is amazing and everybody should read. And we read many books about Humboldt, but I don't think that many people have had the opportunity to really take a book written by Humboldt itself and read it. And it's an amazing experience. I swear to you, you won't be disappointed. I mean, you make a nice cup of tea or coffee, sit in a comfortable chair, and you can read for hours, and it's amazing. And I think we are not really promoting the habit of reading the original works of people who have impacted so much our disciplines, and we should try to do that for many reasons, but mostly because they are very inspiring. The information that they have is precise, is useful. Of course, I mean, it's not, they don't speak about science the way they do now, but I'm really convinced that if more students would read the papers by Humboldt, for example, or the books by Humboldt, they would find inspiration to conduct a lot more research with more passion, because really what you feel when you read Humboldt is passion. And I think that's something that we should encourage and we should cherish a lot more than we do with our work.
Absolutely, absolutely. Somebody else wants to comment on that from the student perspective, either Pedro or Ricardo?
I have something to say. I think it's important also to know the history of ideas. Sometimes we work with hypotheses or with predictions and we don't know where they come from. I like a lot to track those ideas and see who was the first in say that. And I think that that's also very useful to construct a nice thesis and a nice research if you know the history of ideas.
I totally agree, Pedro.
Certainly, certainly, certainly. Ricardo?
Me too. Yes, I think that we need to explore another facet of biodiversity, maybe in very different gradients. In my case, with elevational gradients, I think that historical process originate the patterns, and climatic condition maintain these patterns. But for understanding how this affect in each case, we need to explore functional diversity on phylogenetic diversity and relates with historical process like building mountains or maybe expansion rates or extinction rates. No, I think that is necessary for understand better the elevational gradients and other gradients.
Correct, correct. Great. Okay, do we have any more comments from you guys? Should we start wrapping up and thanking everybody? Yeah. Okay. Okay. So with that, we would like to thank especially our panelists, Professor Jorge Meave, Professor Vázquez, and Ricardo and Pedro, are fine students from the National University and also from INECOL. Ella and Leti for organizing this and inviting me to organize this. And with that, I would only like to share these last couple of slides for reference for people that want to know a little bit more about Humboldt in Mexico. I already shared the expression of Humboldt about the gradient. This is a tableau also that appears in one of the books of Humboldt, The Views of the Cordilleras, which is a representation from his travel from Mexico City to Veracruz. And it's pretty amazing, and I invite you to take a look at it. There's also quite a few books about what Jorge was saying about Humboldt, not from Humboldt, but about Humboldt, you know, as this one, a historian and a geographer in Spanish America, which is not easy to find right now, but maybe you can find it on the internet. There's this one from the Fondo de Cultura Económica, which is Humboldt in Mexico, which is a compilation of essays from the social sciences and biological sciences, which I also invite you to take a look at for people that are interested about Humboldt in Mexico in particular. There's also this one. This is a famous one by Jaime Labastida, which is a Humboldt Ciudadano Universal citizen, and discusses a lot of things about Humboldt, how Humboldt thought about nature and about social groups, especially about this idea of everything is connected, and doing comparative biogeography, we can say now, but actually doing comparisons with different groups and finding common themes and common patterns, which is what we do now in biogeography and macroecology. And there's also this novel about Humboldt in Mexico, which is a fictional novel, but talks about historical characters, Saberes y Delirios from Jose Iturriaga. If people don't know about that one, they can take a look. And there's also this movie, which is some sort of documentary road movie about Humboldt in Mexico, and from the view of the explorer. And you can click on that link, Humboldt in Mexico, and I think you can watch the trailer, or you can try to see if they're gonna show it or somewhere near you. And with that, there's thanks for joining us for the International Humboldt Day. There's more events still during this week, today and tomorrow, and you can find those events at the site. And especially we want to thank Karen Fowler from the IVS and Miguel Matias for organizing this whole event, this whole week about Humboldt, and giving us the opportunity to show some of the Mexican research that is being conducted here and with a Humboldtian view. Leti.
Sandra Noguetu.
Sandra Noguetu, yes. Thank you.
So
Thank
can
you all for...
I barge in just to say one thing? Hola a todos. Muchas gracias por estar aquí y haber escuchado este evento. Me llamo Miguel Matías y represento la Sociedad Internacional de Biogeografía, y os quería agradecer a todos que estuvieron escuchando estos investigadores brillantes de México. Este fue hasta el momento el mayor evento de esta semana.
¿Cómo crees?
Os queríamos agradecer a todos porque van a mostrar del mejor que se hace en México a todo el mundo. Y la razón por qué este evento es en inglés es que a partir de mañana estará en YouTube y los vamos a enseñar a todo el mundo lo que hacen vuestros investigadores. Y so, with that, we just wanted to really thank you all for organizing this. The Humboldt Day was not about just celebrating Humboldt. We did that last year. There was hundreds of events. Humboldt Day this year was to showcase the research that is done all over the world and the people that do that research. And you guys really did a good job. Okay, thank you all. Y muchas gracias.
Gracias, Miguel. Thank you, Karen. Thank you all.
Gracias.
Thank you, all of you.
Thanks, everyone.
Thank you.